Okay, hello, welcome back for another week. So we're carrying on with the AS level. So this is the sort of first part of the A level specification on gene expression. So how the information stored within DNA becomes protein. Now you should already know a little something about how the genetic code works in terms of how the individual bases become mRNA and how they then become how they then get read in and turned into amino acids. Okay, that turns into a protein. So this one is on more about storage and organization. So DNA and chromosomes, obviously the code, the letters work the same regardless of organism, but the way those letters are stored, the way those strands of DNA are stored and kept organized, replicated, so on and so forth, is not necessarily the same across all organisms. Okay, so that's what we're going to cover in this lesson. The bit of syllabus is this bit here, the idea of the difference between prokaryotic and eukaryotic DNA storage. Where else, apart from in the nucleus or the main eukaryotic chromosome, where else do you find DNA? Do you find nucleic acids? What do they do? Where else are they for? Okay, what else are they for? And obviously the fact that in the eukaryotes down here, that a lot of the DNA doesn't actually appear to have the function of coding for protein. So we've told you DNA codes for protein. In eukaryotes, certainly, a lot of that is just not the case. The majority of DNA doesn't code for protein. So more on that as we go through. So let's get stuck straight in with a little overview of what we're talking about here. Okay. So to start with, prokaryotes like bacteria, their DNA is very short. Okay, their DNA is much short. Now, really, for an exam answer, I should be using the word shorter, putting an ER on the end of that. The DNA is shorter than that of eukaryotes. It's circular, right? It's connected up at both ends. It's not wound around any sort of protein or other form of scaffolding structure. You might have these little bits separate from the main bacterial circle of DNA called plasmids, which are tiny little circles of DNA, and you don't find any of this non-coding DNA. Okay, now compare that to eukaryotes, and we're going to go into each of these points in a bit more detail as we go through. DNA is longer, really that should have an on the end, so DNA is longer, right? It's linear, meaning it does have ends, okay? And it's wound around these proteins called histones that then tightly coil to make a chromosome. In some points of its life, they coil so tightly you can actually see them down the microscope. And there is an extensive, large amount of this non-coding DNA, all right, in various places for various reasons, which we'll get to as we go through. So let's start with the structure of a chromosome. So we'll start with eukaryotes. That's you, right? We'll start with us, animals, plants, fungi, the eukaryotes. Now, chromosomes are there all the time, but you can actually only see them during replication. So hopefully you've looked a bit at mitosis, maybe meiosis, and know that during those times, the chromosomes kind of condense down and they wrap really, really, really tightly around these histone proteins so that they can become these visible structures called chromosomes. Okay. Now, at that point in their life, see many previous videos for this particular rant, they are in an unusual state. They've just been through DNA replication, so there's actually twice the physical quantity of DNA there as compared to normal, and they're arranged as sister chromatids, right? These sister chromatids that are absolute copies, photocopies of each other, and they're joined in the middle at the centromere, okay? So DNA is wrapped around these proteins. The proteins are called histones, okay? And how tightly they're wrapped around those histones has a number of functions. So if you didn't have all the DNA in your cell wrapped around histones, it would be a horrible, horrible mess. There's so much of the stuff. There'd be a massive tangle, so it needs to be somehow packaged and stored, particularly during DNA replication, during cellular replication, it needs to be packaged. It's like having a really effective filing cabinet. And also, how tightly a particular bit of DNA is wrapped around its histones determines to an extent whether that DNA is being used or not. Because, of course, if your folder, your file, is closed up really tight, shoved in a really tight filing cabinet, and that filing cabinet's closed, you can't use the instructions in it. Same thing here. If the DNA is wrapped really tightly around histones, then it's not going to get transcribed into RNA and made into protein. The genes that are switched on, genes that are being expressed, are slightly less tightly wrapped around their histones, and more on that in the more advanced topic, which is the gene expression topic at a full A-level standard. So if you're feeling ambitious, go and look at that. It's a lot more in depth on that point. So different eukaryotes, of course, have got different chromosome numbers. So your chromosome number, your base N number is called 23. You've got 23 pairs, so 46 chromosomes in total. Okay, that is not the same for all eukaryotes, not by any stretch of the imagination. So it's most useful, if you're talking generically, to use the letter N just to mean base chromosome number. Then a normal cell has got two lots of that normal number, so that's 2N. Okay, and a sort of cell like a sperm or an egg has just got N. Okay, and you can be quizzed in various exams about those different numbers and be given any species with any base number of chromosomes to work things out from. Okay, so for us, it's 23 and 46. 23 is our N number, 46 is what's called a diploid number. For other organisms, it's different. Okay, so in eukaryotic cells, as we've said, look, there's our nucleus. Okay, you've got the nucleus, you've got the cytoplasm. Everything's nicely compartmentalized. Around the nucleus, you've got nuclear pores that let the mRNA come out. The DNA never actually leaves. Okay, and within the nucleolus, that most dense bit of the nucleus, you've got the production of ribosomes going on because they're actually made of RNA in part as well. But it's not just in the nucleus that you find DNA in eukaryotic cells. There are other parts of the cell that have got DNA as well. The mitochondria and the chloroplast particularly are the main examples. Okay, so mitochondrial DNA there, as you can see. Now, the DNA in both cases here is short, it's circular, it's not got any histones. So hopefully you're thinking there, hang on a minute, that's just like bacteria. Okay, and you'd be right, that is just like bacteria. Okay, so they can independently manufacture their own proteins off that DNA that are obviously involved. In the case of the mitochondria, they're involved in respiration. In the case of chloroplast, they're involved in photosynthesis. The reason they exist and their DNA is like this is because they used to, back in evolutionary history, be free-living organisms. Okay, so much like bacteria, living free and not actually part of a larger eukaryotic cell. And you can actually still see the equivalent of a chloroplast living in the world today. We call them cyanobacteria. Okay, we call them cyanobacteria. So here's our chloroplast compared with a cyanobacterium there, and you can see there's an awful lot of similarities between the two structures and between both of them and any other form of bacteria. And then the picture I put at the bottom there is Darwin, Australia, Shark Bay, Darwin, Australia, and that is a structure called stromatolites. Now, stromatolites are some of the oldest evidence of life we have on the planet, billions and billions of years old. Okay, they were constructed by cyanobacteria, and you can still go and see thin films of cyanobacteria on the outside of these things, laying down minerals as time goes on. Okay, so how are genes arranged in a molecule of DNA? So we've talked about what a chromosome is. There's lots and lots of DNA wrapped around these histones. How are the genes arranged? So what are the sections that are actually doing things? Well, here's your definition. It's a very important definition. Genes are a section of DNA that codes for a particular polypeptide. Okay, codes for a protein. That's our molecular definition of what a gene is. It's a section of DNA that codes for a particular polypeptide. Okay, now each chromosome, each DNA molecule, right, has got lots and lots and lots of genes along it, and as we should know already, the particular position of a gene on a chromosome is called a locus, okay, or many loci, right, and that is conserved. If I looked for your eye colour genes, they would be the same on the same locus and same chromosome as in me, right, as in any other human on the planet. Okay, so just to look at this picture over here that we've got, you can see number one here, look, this is a sister chromatid. So we're looking at this chromosome at that weird time in its life when it's about to separate. All the gene loci on this chromatid will be the same as on this chromatid. In fact, it's just a photocopy, so they will be identical DNA on both. Okay, so 3, for example, might be the loci of one particular gene, 4 might be the loci of the other, of another. And of course, you've got number 2, as you should know there, that's the centromere. Okay, that's the centromere where it's going to split in half so that each new cell ends up with that same complement of genes. Now, on that whole chromosome, if you think about that entire length of DNA, there's actually quite a lot of it, a surprising amount of it, doesn't appear to really do anything. Okay, it might surprise you to know that the amount of DNA that actually codes for protein is between 1 and 2%. Okay? So we think, well, what does the rest do? Okay? Well, there's not actually a lot that codes for proteins. Now, if we're talking about within a gene, there's actually bits that get cut out. And we do a lesson on splicing in a couple of lessons' time. Okay? So non-coding DNA exists, but there's lots and lots and lots of repeating sequences. Okay? This is between our genes. So when we go outside our genes, we've got loads and loads and loads of these areas that don't appear to have a job. Within a gene, there are also bits of DNA that don't appear to have a job. We call them introns. Okay, and you might be thinking, well, why? Why is 98% plus of the DNA in your cells not actually doing anything? And the answer appears to be just accidents of historical legacy. Okay, so you've got all these introns here, 26% estimate here, that basically is the stuff within genes. You've got miscellaneous unique sequences, heterochromatin, and then these things called lines and signs, right? Which are basically these sort of long interspersed sort of regions, some of them repeating, some of them not, some of them unique, some of them not. And the idea goes that over evolutionary time, various mutations have occurred that have actually duplicated bits of DNA, so we end up with repeats. And you might think, well, that's very wasteful. And yes, most of the time it is. Sometimes, however, if you do get a whole gene duplicated that's not the one being used to express protein, then the new duplicate is a little bit freer to experience mutations, produce different proteins, because if it goes wrong, it's not going to kill the individual. And this is one of the suggestions of how all our various types of hemoglobin, right, appeared. So we've got hemoglobin, we've got myoglobin, fetal hemoglobin. They're all coded for by very similar genes, which obviously come from one gene that's been duplicated over evolutionary history. We've also got genes that we don't use anymore, or genes that in us resemble genes that are switched on in other relatives, like chimpanzees and other primates, right? Like the gene for hair all over our bodies, big thick load of hair, is switched off in us and switched on in them. Okay, so this essentially, this sort of mess here, is effectively an unedited accumulation of genes, mutations, errors, duplications, photocopying errors, all that kind of stuff, with no editor looking down and going, man, we don't need that, let's get rid of it, right? For about 4 billion years. Okay? So that is basically what you end up with. So the stuff that's actually being used is only 1 to 2%. Not a sensible system? Sure. But do we survive and get by, therefore pass our genes on? Yes, we do. Okay? You've also got the other stuff in here that's basically DNA that codes for just RNA, and that RNA is used for something, which is another lesson in itself, and also the DNA that regulates the DNA downstream of it, that basically acts as a promoter or on-off switch or things like that. So there's lots of different reasons. The term for this non-coding DNA used to be junk DNA. That's quite, you might see that in a lot of textbooks, but that phrase is kind of on its way out as we sort of discover more where this DNA's come from and what it does. Okay, so moving on, that's our non-coding DNA. So let's some more words for you. Homologous. Now I'm hoping this is all basic genetic stuff you remember from GCSE. Homologous to me means the same. Okay, that's what that prefix means. Normal cells we refer to as normal body cells, or somatic cells as they're called, are referred to as diploid. That means they come in pairs. Okay, humans, as we've said, have got 23 pairs, so 46 in total. Each one of those pairs has come from a paternal source and a maternal source. So in humans, from your mum and from your dad. Okay, homologous literally means the same genes on each chromosome. So this is what we call a human karyotype here. Now we can tell from this picture, if we look at pair 23 down here, an X and a Y, that this is a human male. Okay, so in this case, look, one of these, now bear in mind these are not X-shaped in this picture. These are still chromosomes. They're not X-shaped and they're not joined in the middle. So these are pairs of chromosomes. Right, so that one there might be maternal, that one there might be paternal. As you can see, look, the banding patterns when we stained them are the same. So that's what we mean by homologous. The loci and the particular genes are the same on each one. So they're the same genes, so at the same place on each chromosome 1 or whatever, you might have the same gene for eye colour, or whatever it might be, but they won't necessarily be the same version of the gene. And that's where this word allele comes in, that you surely would have heard of at GCSE. Okay, so the same genes at the same loci, not necessarily the same version of the same gene. Okay, so haploid cells are cells which only have one of each chromosome. And most commonly, we're talking about gametes here: sperm, pollen, egg, or anything used for reproduction. Not always the case. There are organisms that that reproduce and just have haploid cells just grow. So haploid cells going through mitosis and becoming the adult organism. So things like ferns and mosses go through this thing called generational alternation, where one generation's haploid, one generation's diploid, one generation's haploid, one generation's diploid, which seems to work for them quite well. But for you, for us, for most eukaryotes that you'll come across, gametes means sex cells, reproductive cells. Okay? So on this allele, again, I'm hoping that you are well familiar with this term. An allele is a version of a gene in a specific locus. For example, eye colour is the famous example that every GCSE biology teacher uses. Cystic fibrosis is another one you should have come across at GCSE. So those are particular versions of particular genes, a particular allele, a particular loci. Blood type, another one you might have heard of. You either have an allele for the A antigen, or the B antigen, or the allele for the O antigen, which is no antigen, or a combination of those ones. If you've got two O alleles, you'll produce no antigens on the surface of your blood. That makes you blood type O. If you've got one A and an O, or two A's, you'll produce A antigens, your blood type A. And if you've got one of each, an A and a B, your blood type AB, because you produce both antigens. Okay, so that's another example you might have heard of. Mutations, of course, generate new alleles. So if anything happens during your life that you can then pass on, some of them are good, right? Most of them are bad. For a lot of them, it's not that simple. So things like cystic fibrosis, obviously very bad if you get two of them, but in a heterozygous form, there's good evidence that it actually gives you some protective advantage against the disease cholera, which is apparently why the gene frequency moved up to such a high level in populations of people who were early city dwellers, like Western Europeans. Okay, but again, that gets into evolution. You go and look into that in your own time. Okay, right, that's your eukaryotes done. Let's switch over to prokaryotes now. So we're talking about bacteria primarily here. Bacteria, no membrane-bound organelles. You should know that from the beginning of your AS level. Therefore, where do all the cellular reactions occur? Well, either they're just in the cytoplasm, or if it's a reaction that requires a membrane, like respiration and photosynthesis, they tend to use the inner, the membranes and the space between their cell wall and their cell membrane. These sort of folded bits called mesosomes, which we have only relatively recently discovered that's what they're for. Okay, their DNA is in one big circle. Okay, one large circular chromosome, and of course we've already mentioned they have these other smaller circles called plasmids. Please don't say that all bacterial DNA is plasmids, right? Because it isn't. That's a very common mistake, okay, at AS level. You have the majority of the DNA in this big circle, we call it a bacterial chromosome, right? That big circle, and then little tiny extra bits called plasmids. Just because it's a circle doesn't mean it's a plasmid, okay? Not all circles of DNA are plasmids. Right? That's a very common mistake. So you've got one large circular chromosome and then additional plasmids. So on plasmids, here's the what you need to know. They've got these small loops of DNA called plasmids. Why are they there? Well, they replicate independently and they can be swapped. Now, I don't know if you've come across this before, but this is the closest you'll get to bacteria sex. Right? We call it horizontal gene transfer in bacteria. The idea that these plasmids can be photocopied like a little tape, right, like reeling off a tape, and pass through what's called a conjugation tube. Okay, you might remember learning about the structure of the pilus on the outside of the bacteria. Now these pili can be used to attach to another cell, make a conjugal tube here, right? They actually pull themselves right close together, and they get membrane to membrane contact and pass a plasmid through. And it means that useful genes can be transferred horizontally, not just down the generations, from different bacteria to different bacteria. And the classic example of this is antibiotic resistance. This is how antibiotic resistance has spread so rapidly across all sorts of different species So it doesn't just crop up in Staphylococcus, it also you find it in various Bacillus species, various Streptococcus, all these different various E. coli's, all those kind of stuff, right? Because they can not only share genes between their own species, but they can actually conjugate with different species. Absolute filth, right? They're disgusting little creatures. Okay, so here we have a sort of labelled version of what's going on. So that's the plasmid of the non-conjugative type here, look. This plasmid is about to pass one strand of DNA into this pilus. Right, the pilus conducts the plasmid to the recipient bacterium, right through there. Okay, and in there you've then got the plasmid of the conjugative type. Okay, so that's basically how it works. Now I'm going to show you some pictures here. So if you're of a nervous disposition and can't be bearing to watch these filthy creatures doing what they do, it is just nature. It doesn't make it nice, right? But I will show you here some of this absolute bacterial filth. Look at those. I apologise, but this is